Hi everyone. So we left off on the antigen features. Remember, the antigens are what you would find as part of a virus or a bacteria, but it is the molecule that will start or be recognized by our body as something that needs to be fought. So if you look over here, we have an epitope, which is just a part of an antigen that is shaped, in this case, like a triangle, and we can see that we have a T cell receptor, so a T cell that actually has an antigen binding site that matches the shape. So this particular T cell is only going to recognize a specific epitope that looks like this. This is true for B cells as well. They have two antigen binding sites on their receptors, but again, each B cell is going to be specific for one antigen, and in this case, it's a circular shaped one. So remember, I talked about in the last lecture that we have a library full of different types of B and T cells, all with different receptors. So each one of them, each B cell and T cell, would have a different antigen binding site in our library, just waiting to be affected by an epitope or an antigen. So we'll probably come back to that. I'm probably going to do a little drawing on a tablet for you guys that I'll also release later this week. So when we think about the antigen features, so as we think about these antigens coming into our body, whether they're part of a cell wall of a bacterial cell or they're the spike of a virus, B and T cells, each B and T cell will have thousands of receptors on their surface. But keep in mind that for each cell, all of the receptors on that surface will only recognize the same epitope, one epitope. So again, it's a big library with lots of diversity, but very few of each type. So when that B or T cell binds to a specific epitope of an antigen, that's when the cell becomes activated. And so if there's one T cell that recognizes that triangle-shaped antigen, when it actually binds to that specific epitope of the antigen, it's going to become activated. And when it becomes activated, it will begin to go under what is called clonal expansion. It's basically going to make an army of clones, T cells, that recognize triangle antigens. So we don't have to have a lot of each type of T cell or B cell. We just have to have one that can recognize any antigen that comes in, and then those can be made into clones when needed. So some of those clones will develop into these effector cells, the ones that actually rid the body of the antigen, while others become that memory cell, so that if we face that antigen again, our body will react more quickly. So activated B cells will differentiate into those effector cells that are called plasma cells. So B cells become plasma cells, and these plasma cells will make antibodies. These look like the B cell receptors. Remember, if I go back really quickly, the B cell receptors have this kind of Y shape when they're attached to a B cell, and they have two places to bind an antigen. When this differentiates into a plasma cell, it will make antibodies, which are just these B cell receptors that are then secreted by themselves. So that's what happens when the B cell becomes activated. We'll talk about what those antibodies do when they're in our blood and they're circulating in our body and how they go about taking care of the antigens. T cell activation, remember, leads to differentiation into very diverse effector cell lineages, and we'll talk about what those are. So T cells are interesting. The two that we're really going to concentrate are the helper T cells and the cytotoxic T cells. So the cytotoxic T cells can actually directly destroy a cell that is infected with like a virus or a bacteria, or a cancerous cell, so a cell that is growing uncontrolled. T cells will actually destroy those. T helper cells are a little bit different, and I think of T helper cells as kind of the generals in an army. They don't actually directly seek and destroy the invaders, but what they do is they basically delegate or order all of the adaptive immune response to become stimulated and to begin to fight the disease. So interestingly enough, if you know anything about HIV or AIDS, HIV, the virus, it actually attacks these helper T cells. That is the tropism that it has is for helper T cells. And so the reason why HIV is so dangerous and makes us so immunocompromised is that because it infects our helper T cells, it will actually wipe out this whole population. And when that whole population of helper T cells wipes out, it means we lose this coordination of adaptive immune response. That's why there's immunosuppressed. So just a little bit of interesting to think about the role these cells play. So the way that we tell our cytotoxic T cells from our helper cells is the fact that they have proteins on the outside of them that are called cluster of differentiation proteins. We call them CD. For helper T cells, they have CD4 as their proteins, and for cytotoxic T cells, they have CD8. So you may have heard with HIV, we look for a CD4 count, and that's to tell us how many helper T cells somebody has. So our helper T cells are the most abundant of our T cells. Remember, they have that CD4 protein on the outside of them. It's how we know what they are. And their whole job is to help coordinate the adaptive immune response. And they do this by releasing cytokines, those chemical signals. They also activate other white blood cells, and that's usually through cytokines, these B cells, macrophages, and cytotoxic T cells. Remember, I said they're the generals. They're sending out orders to all these other white blood cells to start fighting an infection. They are the main cell that organizes both the cellular and humoral branches of adaptive immunity. And when they activate, they can be differentiated into a variety of subclasses that each have different functions. So think of those helper T cells, though, as really running the immune system at this point. So this gives you an example of how this might happen. We have our CD4 helper T cell. When it is activated, it goes through proliferation. It can do helper T cell 1. These can stimulate cytotoxic T cells. It can differentiate into T helper cells 2. These will stimulate B cells, so get that humoral response going. And we can have T regulatory cells, and these are T cells that basically tell the immune system that once they've gotten rid of the antigen, they can calm down. So that's kind of our helper T cells. Our cytotoxic T cells, remember, are the ones that directly destroy cells that are virus infected. Maybe they're damaged, or they are a foreign or transplanted cell. So think about things like transplanted organs. These are the cells that we would think of that could destroy those. Or they can also destroy cancer cells. Remember, I talked about that cluster of differentiation, the different proteins. These type of T cells show CD8. That's how we know that they're cytotoxic T cells. So when we think of the T and B cells, remember I talked about the idea that they all have receptors. Each T cell and each B cell has receptors for a specific antigen. So the idea is, how do we make sure that our T and B cells have enough receptors to be able to recognize a wide variety of antigens? Remember, it's that whole library, and I'll do a quick drawing of this that I'll put out this week. And they do this through something called gene shuffling. Basically, they have a random process that gives the shape of the receptors on each of these T and B cells. And so what this does is it allows us to have this big library. One of the things, though, that we have to look out for is that because it can make a receptor for basically any shape, we have to make sure that if a T or B cell has a receptor that would bind to a normal body cell, so maybe a muscle cell or skin cell, we have to get rid of those. We don't want ones in our library that recognize our tissue, or we're going to have an immune response to our tissue. So what we have is we have a mechanism to screen for those that recognize immune cells, and we look for ones that are tolerant of self, meaning that they will not attack our cells. So that's a really important thing, is to make sure that we don't have that. And that's sometimes what happens where we have autoimmune disorders, is that the body is actually making immune cells that are not self-tolerant. So the way that we look at this, remember, the T cells are going to mature in the thymus, T for thymus. B cells will mature in the bone marrow, so B for bone. And what we look for is, in the first with T cells, we're thinking about the idea that they have to have an antigen presented to them, and that's done by this major histocompatibility protein. We'll talk about that later in the chapter. So does it recognize that? Yes, that's good. We want to make sure that it recognizes a cell that presents it to us. If it doesn't represent that, we get rid of them. But if we can recognize these antigen-presenting cells, that's good. But then, are we self-reactive? Do we react to a self cell? If the answer is yes, then we want to get rid of you. We don't want to have something that recognizes us as foreign. If the answer is no, that's great. This can now be circulating and looking for antigens. And here with the B cells, can it make antibodies to self-antigens? If the answer is yes, we don't want that because we don't want our B cells reacting to us. We only want ones that don't react to self-antigens, and those we keep. So that's that idea of self-tolerance. I just mentioned that for T cells, they have to be able to recognize this major histocompatibility complex proteins. These are basically self-proteins that we see that allow a T cell to recognize that basically a friend is presenting them with an antigen. And so as the T cell matures in the thymus, it must be able to recognize these self-MHC proteins, but it cannot attack self cells. So that's the kind of difference there. And I'll also do a short lecture on the tablet for this. So the mechanism for B cells was a little bit different in that it occurs in the bone marrows, and it just needs to make sure that its future antibodies won't react with self-antigens and damage host tissues. Those T and B cells that can't tolerate self cells or recognize self cells as foreign are then signaled to undergo apoptosis, which means cell death. So any cells that would recognize the self cells as foreign, so any of our T and B cells that would recognize our cells as foreign, must be gotten rid of so that we don't have a reaction to our cells. So this is a quick kind of summary of the difference between the T cells and the B cells. We know T cells make up the cellular branch, B cells make up the humoral. The ones that we're really looking at are the helper T cells, or CD4, and the cytotoxic T cells, or CD8. Remember, they mature in the thymus because it starts with a T, and they're T cells. These are mainly found in our lymphatic tissues, but can be found in low levels in our blood. They have T cell receptors that recognize the antigens. They require an antigen-presenting cell, and they will make a memory cell. We know that they have these MHC surface. We'll talk about that a little bit more when we get into the cellular branch. And they're not considered antigen-presenting cells, but they need antigen-presenting cells to see the antigen. B cells, they will make plasma cells that can make antibodies. They mature in the bone marrow. Remember, B cells, bone marrow. They're again found mainly in the lymphatic tissue, although low levels are in circulation. They have B cell receptors, and when they're activated, they will secrete, their plasma cells will secrete antibodies. They do not have to have an antigen-presenting cell present the antigen to them. They can interact with it directly. They will make memory cells, and they can show both MHC1 and MHC2 proteins on their cell surface, and they can also act as an antigen-presenting cell, so they can present antigens to T cells. So, some checking in. You should be able to name the two responses of the adaptive immunity and which one is associated with T cells versus B cells. You should name the two key aspects, the key words of adaptive immunity that I talked about. You should know what makes a good antigen versus a poor antigen. Be able to define self-tolerance and apoptosis. So that's the end of 12.1. If you have any questions, again, please let me know. And I will try to make sure to get a picture of kind of the receptors and what I mean about building that library on my tablet. So I'll try to post that as well. Please contact me if you have any questions. Thanks.